So now we're going to talk about an overview of the cell and glucose and then get into a discussion about cellular respiration. With a eukaryotic cell, we have a plasma membrane, a nucleus, and then we have mitochondria, which have a double membrane. With a bacterial cell, prokaryotic cell, there's no mitochondria, there's no nucleus, there's just a little region inside of the cell where you'd have some DNA. Then you have glucose. So glucose looks something like this. And you get the picture of glucose. So when we look at glucose, glucose has to be able to get inside of the cell, whether it's a eukaryotic cell or a prokaryotic cell, in order to start the process of breaking it apart to make energy. So in the process of glycolysis, and even if we just look at this term, glycolysis. So there's that word lysis again. Lysis means to break apart. We start with glucose, and we're trying to break it apart. And there's actually 10 steps involved in glycolysis. I'm only going to have you remember the first few steps because those are key rate-limiting steps in this whole process. And they're committed, irreversible steps. So once they happen, there's no going back. The first step, when we have our glucose, glucose gets brought into the cell, and inside of the cytosol, which is where this happens, whether it's a eukaryotic or prokaryotic cell, glucose is converted to glucose 6-phosphate. Going back to this picture here, if we get rid of what glucose looks like, and I'm going to draw it a little bit bigger now. In organic chemistry, each carbon is numbered. So if we look at this structure, each one of these corners is actually a carbon. So this is carbon 1, carbon 2, carbon 3, carbon 4, carbon 5, and carbon 6. And the chemical formula for glucose is C6H12O6 because there's 6 oxygen, 12 hydrogen, and 6 carbon. Well, we just labeled the 6 carbons, and then the oxygens there's 1, 2, 3, 4, 5, 6, and then the hydrogens you can fill in from there and count the hydrogens. They're all there. So the first step of glycolysis, when we take glucose and convert it into glucose 6-phosphate, what we're doing is we're adding a phosphate to the 6th position carbon. Why do we want to do that? Well, adding a phosphate to it, once it's inside of the cell here, traps it in the cell. Now this thing can't escape. It's stuck inside the cell. So our first step is really important. Traps it inside the cell. Plus, it affects the concentration. So if you know biological systems like to have an equal concentration inside of the membrane as outside of the membrane, if we trap it inside the cell as glucose 6-phosphate, that's different from just straight up glucose in the surrounding environment. So it allows it to stay inside the cell. Then we convert it to fructose 6-phosphate. And let me actually get rid of that. Go back to my black pen. Fructose 6-phosphate. The purpose of glycolysis is to break glucose in half. We want to have this thing looking symmetrical, so when we break it in half, we can do so symmetrically. Well, this doesn't look very symmetrical. When we look at this structure now, we see this random phosphate hanging off over here on the side, and we want to make this look a little bit better. So we take glucose 6-phosphate and then we convert it into fructose 6-phosphate. Now fructose is actually a pentamer. And we end up with something that looks like this. So we have the phosphate attached to that carbon still, but now the carbons are rearranged in a little different ways. So now this structure is fructose. You can see if we want to rip this directly in half, well, this side has a phosphate, so this side should probably have a phosphate. And that's exactly what we do. The very next step here, fructose 6-phosphate, we go fructose 1,6-biphosphate. Sometimes it's called bisphosphate or diphosphate, same thing. Because we number our carbons 1, 2, 3, 4, 5, 6, the first carbon position now is this one because we've rearranged it in fructose. So this would be carbon 2, 3, 4, 5, and 6. So if we want to have fructose 1,6-biphosphate, we would add a phosphate to the first carbon because we already have a phosphate in the 6th position carbon. So we would add it here. Immediately following that, we rip it in half. So now glucose is ripped in half, and through a series of a bunch of other steps, we end up with 2 molecules of pyruvate. And in this process, we made 4 ATP, but then we ended up using 2 ATP. So overall, there's a net gain of 2 ATP. So if you make 4, but you lose 2, then you have 2 left. So in the end of this process, we ended up with 2 ATP. For exam purposes, the details that I put on this slide, you're not going to find that on an exam. This is just to give you a visualization of what's actually happening. Some people are visual learners. They like to see, well, what's actually happening to this molecule in the cell? This helps explain that a little bit better. The details on this slide, you definitely do need to know. So moving on to the Krebs cycle now. So the Krebs cycle is going to happen in the mitochondrial matrix if it's a eukaryotic cell. Or just in the cytosol if it's a prokaryotic cell. Before we can enter into the Krebs cycle, we start with pyruvate. Then we have this gigantic complex known as like pyruvate dehydrogenase, but this is huge enzyme complex that converts pyruvate to acetyl-CoA. So CoA is this large group that just carries around this acetyl group. Once it's converted to acetyl-CoA, now it can enter into the Krebs cycle. And they call it the Krebs cycle because as long as you're pumping in acetyl-CoA into the system, it can continue going around the circle. Acetyl-CoA combines with acetylacetate, or oxaloacetate, and that starts this process through all of these different enzyme complexes. This cycle is so important because this is the component of cellular respiration where you can have inputs of amino acids. So amino acids can jump in here to help make energy. Lipids can jump in here to help make energy. Components of this can go away to go make lipids or go away to go make amino acids. So this is kind of the central hub of all of the metabolism, this Krebs cycle. You do not need to know each one of the enzyme pathways associated with this because it's interconnected to so many other things. And this isn't a metabolism class, this is a microbiology class. So the key component that you do need to know is the fact that the Krebs cycle releases carbon dioxide as a waste, releases 2 ATP, and then gives off these electrons and hydrogen ions. And these are going to be transported to the next step by an electron transporter known as NAD. And NAD can transport 1 hydrogen at a time. And this is really a shuttle that's going to shuttle back and forth between the Krebs cycle and the electron transport chain. The carbon dioxide again leaves as waste, and the ATP, that's a good thing to have around in the cell. Where did this carbon dioxide come from? Well, if we go back to glucose here. This is glucose, C6H12O6. When we broke it in half to make pyruvate, pyruvate still consists of carbon, hydrogen, and oxygen. The carbon and oxygen combine to make carbon dioxide. And then the hydrogen ions go on to the electron transport chain. So now just by looking at this formula here, you can see where the carbon dioxide came from. If you are a eukaryotic organism like a person, this carbon dioxide waste can accumulate inside of the body and become acidic. It can turn into carbonic acid, and that's acidic. So we have to breathe this out. We have to get this out through our respiratory system. So our bloodstream will deliver it to our alveoli, and it's going to escape the bloodstream at the alveoli. As a bacterial cell, you just spit it out of your cell membrane, and then it's in the environment, and it's not your problem anymore. So coming back to the Krebs cycle, we gave off this waste product, we've got ATP, and then we've got these things that are carried to the next part of the process by this NAD transporter. Now we have the electron transport chain. And the electron transport chain is going to happen between the two membranes of the mitochondria in eukaryotic cells. And in prokaryotic cells, it's just going to happen between the layers of the cell wall. So the cell membrane to the cell wall. So the electron transport chain consists of this large spanning intermembrane complex. So if we have a phospholipid bilayer here, so this is a double membrane structure, and then we have a phospholipid bilayer here, so it's another double membrane structure. And then there's a space between the two membranes, this intermembrane space. Well, you've got these large complexes that span inside of here. And there's 4 complexes, and then you've got this giant ATP synthase enzyme. Which I'll abbreviate ATPase. So NAD is busy delivering hydrogens that travel through these complexes. And then there's electrons that are bouncing around externally from complex to complex. FAD is also involved in these complexes, and FAD can transport 2 hydrogens. So now you've got an accumulation of hydrogen ions in this intermembrane space. And they're super concentrated, and if you know anything about acids, acids have a high hydrogen ion concentration. So this is really acidic and volatile, and we don't want a whole bunch of concentrated positively charged ions within this intermembrane space. Number 1, they're concentrated, which means they don't like being by each other. Number 2, they're all positively charged. If you have too much positivity, they're going to repel each other. So we can't have all of that positive charge in that membrane. So they want to get away from each other, and the only way that they can get away from each other is by going down their concentration gradient. And there's only one channel that lets them through, this ATPase channel. So as the hydrogens go through down their concentration gradient, well, now they're back inside of the cell. So this is the cytosol. So as the hydrogen ions have flowed through this channel, it causes a rotation. And this thing will rotate and rotate and rotate. And that rotation takes ADP, adds a phosphate to make ATP. Every time a hydrogen goes through, it provides momentum for the rotation of this, and that momentum allows for these two to come together to make ATP. 32 to 34 ATP can be generated this way. So now as the hydrogens have gone through, they need to combine with something because they're still acidic and there's still a lot of them, and you want to neutralize these. So what we do is we bring oxygen in, and as a human, you're going to breathe in the oxygen. As a bacterial cell, you just suck it up from the environment. So hydrogen will then combine with oxygen to make water. So oxygen in this situation is known as a terminal electron acceptor. And the reason why we call it aerobic respiration is because oxygen has to do with being aerobic, is our terminal electron acceptor. So this kind of defines the process. Sometimes bad things can happen, and you can have the wrong combination of hydrogens and oxygens, and you end up producing hydrogen peroxide. Catalase destroys that hydrogen peroxide. So if you're aerobic, you're likely going to be producing catalase to help neutralize this. I just want to talk a little bit more about this because we talked about it with the enzymes, but I want to give you another clinical application to why this stuff is so important. Lactobacillus can be found inside the digestive tract and inside of the vagina. My master's thesis happened to focus on lactobacillus in the vagina and the protective capabilities of lactobacillus, like how this microorganism can help protect our vagina against pathogens. So, if you're completely colonized with a good kind of lactobacillus, like Lactobacillus crispatus, for example, that Lactobacillus crispatus physically blocks the penetration of pathogens, but also produces toxic compounds that kill off pathogens. One of the toxic compounds is known as a bacteriocin. It's kind of like an antibiotic-like substance. Another toxic compound is lactic acid, which causes the pH of the vagina to drop. So with your vagina being really acidic, you're not going to have an overgrowth of pathogens because most pathogens don't like swimming in a pool of acid. That's why if the pH starts to increase, you're predisposed to getting bacterial vaginosis or bacterial vaginitis. But another toxic compound that lactobacilli produce is hydrogen peroxide. Hydrogen peroxide has been shown to interrupt the replication of the HIV, like human immunodeficiency virus. Hydrogen peroxide alone. This is huge. This means that if you're well colonized with lactobacillus, the hydrogen peroxide produced by that bacterium has the capability to interrupt pathogens. So not only is it an accidental combination of these and we produce catalase to get rid of it and so forth, if we let it sit inside of areas where bacteria are beneficial, it can actually kill off pathogens. So anyways, that was kind of a side note about the importance of this formation of this hydrogen peroxide. But let's wrap up here by talking about aerobic or anaerobic respiration and fermentation. So anaerobic respiration is simple. You're going to go through all of these steps. You go through glycolysis, you go through the Krebs cycle, and you go through the electron transport chain. Except when you get to the very end, that electron acceptor is not oxygen, it's something else. Usually the something else is going to be sulfur. So in anaerobic respiration, you use something other than oxygen as your terminal electron acceptor. Usually it's sulfur. And if it is sulfur, you're going to produce H2S. This H2S can be detected by adding certain substances to it, and it forms a black precipitate. And there's a couple tests that we'll do in the course of the semester that you'll see that black precipitate form. Now with fermentation, fermentation is different altogether. Fermentation is glycolysis only. And then you end up producing an alcohol or an acid as a waste product. And like I mentioned earlier, alcohol is going to be produced by yeasts predominantly, and the acids are going to be produced by different types of yeast as well, but then mostly bacteria. So bacteria have the capability of producing different types of acids, and we can test for acid production and what type of acid is being produced, and that helps us diagnose infections. Here is an overview of catabolism. So it shows aerobic respiration, glycolysis, Krebs cycle, and the electron transport chain. Anaerobic respiration, glycolysis, Krebs cycle, and the electron transport chain. And then fermentation. And fermentation is just glycolysis, which leads to the production of an acid or an alcohol. This is a very great summary table that I highly recommend you review before the exam.